Okay, let's get started. I think we have a full program. My name is Benjamin Peters. I'm a member of the program committee here, and I'm very excited to announce the second GAC here, Attending to Errors in Predictive Coding, a collaborative community experiment through the Open Scope program. Very briefly, generative adversarial collaborations, I've already said that for the first one, is one of the community-organized events of CCN, a very innovative program. And the idea is that science should advance through an exchange between theories and ideas, but often it happens that, I don't know, like a certain discipline stays with themselves and there's an alternative theories, they stay with themselves and they acknowledge each other, but they don't really talk the differences out. And that hinders progress. And so that's where GACs come in, where people meet with different alternative theories and they want to collaborate and then resolve these differences. And this often involves first finding common language, and I think you started doing that, defining terms precisely, developing a shared reality, as we heard on the first day, and of course with the purpose to accelerate science. We recently put out this preprint describing this project here from CCN, also some guidelines if anybody wants to implement this in a new field, how to organize this. And please go to the GAC webpage where you find all the previous GACs, all the recordings of the workshops. And you'll also find a link to today's schedule where you already find a half-written paper, which I find amazing, so check this out. You'll find 7 preprints. There will be an 8th one soon of the 15 GACs that have been organized over the years. And I can say GACs are great. I participated in one myself. I think it was quite transformative for myself, and I really enjoyed the deep discussions that necessarily come up if you sit together and really want to understand the other side's perspective. It's a lot of work, but it's very rewarding. So I can absolutely recommend considering submitting one next year as well. So please consider submitting a GAC next year if you're intrigued. And so without further ado, let's welcome the organizers on the stage with a round of applause.
All right. Thank you, everyone, for coming to our workshop on predictive coding. As you can guess, I'm only one of the organizers. I'm Colleen Gillen. I'm a postdoc at Imperial College London with Claudia Klopath. And I'm one of the co-organizers of this workshop with Michael Berry and Jérôme Lecoq, whom you'll be hearing from later in the workshop. But this has already been truly a very collaborative effort. All of our speakers have been very involved in preparing this workshop. So I think it's going to be a great event that we have for you guys today. I hope you guys enjoy it. I'll give you a brief overview of what the structure will be. So the idea with this workshop is to bring together the community of people who are interested in predictive coding as a theory of how our brain processes sensory information in the brain. And to get a lay of the land, what is the current state of, from a theoretical and an experimental point of view? And what are the key experiments that we need to be running to really advance the field, to move the needle, and to push further as a group and not all in our separate silos, as Benjamin was alluding to. And with this in mind, we've actually already been putting together a position paper, one that everyone is welcome to come be a part of. Sorry, I should only stay towards the mic. This is the position paper that we've already been putting together over here. And as you'll see, we have all these details on contributions. So if you have expertise in the field, trainee, PI alike, we would welcome your contribution. Come join us. We really want people who have expertise in all the different aspects of predictive coding to be a part of this project. And there are already many, many comments. And we also have a section that'll sort of be the focus today on experimental proposals. And that's where we hope to start to really build out some concrete proposals for people to go and run their experiments and move the field forward. So for our workshop, getting back to that slide. We'll be starting with a section of talks on the theory side of predictive coding, followed by talks on the experimental side. And there are sort of these 6 pillars that we've identified for approaching predictive coding. So one is the different types of errors that you can introduce experimentally to study predictive coding. The other is the role of hierarchy and layers in the brain in implementing predictive coding. Another pillar are the types of prediction and error signals that have been observed experimentally and reproduced in theoretical models. And then we have the role of inhibitory circuits in apical dendrites. And lastly, the types of learning and plasticity mechanisms that could be underlying predictive coding. So you'll see these themes pop out during the different talks. Now, these two sets of talks will be followed by the most important part of the GAC, which is going to be the discussion. So a few of us will start by putting forward 3 proposals for experiments that we've come up with as a community. But that's where we'd really like to open the floor up for discussion for others to share their ideas of what we should be doing next. So during the talks, don't hesitate to start thinking about your ideas, and then there'll be 2 mics here for people to come contribute at that stage. One of these proposals will be run in the OpenScope program. Jérôme's going to come up to tell you guys a little bit about that. So,
All right, so OpenScope, briefly, I'm going to make my spiel so you can learn about it on the website at the top. We are the first astronomical brain observatory in neuroscience, I believe. So we run new experiments at the Allen Institute that are proposed by the community. We operate on a yearly basis. We release our data to a Dandy archive. You can already see some of the data, but we have released in the past. And we are funded by the Brain Initiative. So we are truly a community brain observatory. So this workshop for me is really dear to me because what we are trying to do here is bring the community together so that we can design an experiment in the open. This is something that the field has, I think, rarely done, and which could be really impactful that's what I believe. And so I want to thank the OpenScope steering committee, notably Satra and Mackenzie and Nick Steinmetz, that really pushed us to do that, what we're doing today. Without further ado, let's continue.
All right, I'm almost done the intro. So we do have a fairly short GAC in our 45 minutes. The talks will be 8 minutes each, and we have 2 minutes for questions, but it's really mostly buffer time. So we'll only take questions if you have a concrete clarification question. Please keep your more extended questions for the end. And then afterwards, we would like to continue the discussion. So if anyone wants to join, we'll be going to Nako Taco 8 minutes away to continue chatting and get your input. And at any point, you can scan the QR code to go see our position paper. It'll appear on numerous slides.
Hi, so I'm Fabian. I'm a postdoc in the lab of Viola Priesemann in Göttingen. And I'm going to talk a bit about a theory that we've been developing about how predictive coding could also be implemented in a different way than typically assumed with error computations in the dendrites. And the basic idea is very simple. So before, as Claudia also talked about a bit, people commonly assumed the cellular hypothesis where the error is computed in specific neurons, and we kind of shift this picture or this view and say that this error could also be computed in the dendrites. And there's a few reasons why we assume this, or why we think this is a good way of looking at this theory. And I can go through the details, of course, but I want to look at 2 important questions, which are, first of all, why do we consider the dendritic hypothesis? Why do you think it's important to look at that from a theoretical perspective? And then what are the central predictions that this theory generates? And the short answer to the first question is, why do we consider it? Because it is at the moment the only feasible mapping of learning and predictive coding to spiking neurons. And to explain it a bit, what I mean with that, I don't want to contradict Claudia's results. I mean something else. And I have to start for that from the beginning of the theory. And that is that predictive coding is a story based on Marsh 3 levels. So I think that's a really nice part about predictive coding, that it really starts from a computational goal, and then over an algorithm tries to map this implementation of the computational goal to the biological implementation. So we get a nice connection between the biology and the function in the end. And as many of you may know, the computational goal in predictive coding, at least how it was, for example, proposed by Rao and Ballar, is to basically try to understand what objects outside in the world are. And so what is assumed is that there's a world in itself that is external of the observer, and we only have the image that we observe, for example, and try to infer back from that what are the objects that constitute this observation. And not only do we have to infer these objects, we also have to learn what are these objects in the first place. So before we, let's say at the start of our life, we don't know what objects there possibly are, so we have to also learn from our observations what categories there are. And the way that predictive coding approaches this problem is that it first defines a generative model, which you can see on the left, which is basically just a probability distribution. And I can't go through the math and the derivations, but in the end, we can derive update rules for abstract mathematical quantities, which represent the predictions or representations. And we can get that for both the inference and the learning part. And what we find is that both inference and learning depend on an error that we can compute by comparing activities in the levels. Yeah. So now we have this algorithm and we want to map it to the biological implementation. And as I said, the classical way to do that is to take this error and map it directly to the activity of individual neurons. And in that case, the learning rule becomes this Hebbian learning or plasticity rule that just multiplies the error and the representation or prediction neuron activity. And the problem is, it is not clear how to implement this learning with spiky neurons. So this update rule basically tells us how we update from the error neuron the weights to the prediction neuron, and it's not clear how this is feasible if you exchange the error neurons through spiking neurons. And the reason is basically that these errors can be both positive and negative, but the neural activity itself can only be positive. And what Claudia also talked about is a common way how people think this could be resolved is that we basically take two types of neurons, the positive and the negative error neurons that code for the positive and negative part, respectively. But the problem with this idea is if we now look at the plasticity rule from the error to the prediction neurons, these become non-local because in this case, the plasticity rule suddenly depends on both these positive and negative errors, but for a single sign, and so they depend on three factors, and it's not really known how to get this plasticity rule local again in the end. So this was one of the problems that we saw, and what we then proposed is that essentially the same learning algorithm could also be implemented if these neurons, if these errors are computed in the dendrites, and in this case the learning rule becomes a voltage-dependent plasticity rule, which depends on the local voltages in the dendrites. And here, of course, you don't have this problem that you cannot code for negative error neurons because just a drop in the voltage can show this negative error. Okay, so that's the main reason why we consider this from a theoretical perspective. And now I want to talk a bit about the central predictions. I cannot go through all the things that this theory predicts, but I want to focus mostly on this first part, namely the different roles for apical and basal dendrites, which we consider in the theory. But there are other also very interesting predictions, for example, for the lateral inhibition between neurons, or as I said, also the plasticity. And what we basically discuss in our models is that there could be two different modes of error computation for the basal and apical dendrites. Okay, one of them is, both have been proposed by others before in a similar way. One of them is a spike-based error computation between the apical dendrite, which is predictive for the somatic activity, and then we compare basically these two activities to compute the error, and the other is a balance-based error computation, where we compare excitation and inhibition between excitation and inhibition in the basal dendrites, which also has been, for example, proposed by Deneve and Machans. You see that there's no SOM and VIP neurons in the circuit, which are also really important, most likely. We think these are important for attention gating, but not for the central computation in the circuit. But there's also other models by, for example, Sacramento that also employed dendrites, but don't propose these roles. And there's some indications that this makes sense. So what people have been observed is that there's different learning rules in apical and basal dendrites. And what also Colleen showed with others together is that it's at least compatible with the idea that these apical dendrites become predictive and the basal become more balanced through learning. And yeah, there's still a lot of things to be tested. For example, that the apical dendrite really becomes predictive for the soma is not clear yet. Okay. Oh, sorry. Sorry, these are the old slides. So to summarize, the dendritic hypothesis enables learning and predictive coding with striking neurons, which is very important from a theoretical perspective. And there are several predictions for how neurons could, on a cellular basis, perform this dendritic computation, this dendritic predictive computation. And you can also look at our review if you're interested in more details. And with that, I want to thank you. And if there's no questions, I did most of this work together with Lucas, another postdoc in the lab, and Viola. But yeah, thank you.
Okay. Hello, everyone. I'm Michael Berry. I'm a professor at Princeton University. And I'm going to try to delve a little more broadly into this question: why does the brain really want to make predictions, and propose some different sorts of experiments to test some of the conclusions. So I want to start with just a very brief early history of predictive coding, because I think it lays out a set of ideas that have been very influential. So the idea really starts in the 1950s with Barlow and Atneve, and they're sort of thinking about retinal ganglion cells with center surround receptive fields, and what they realized is that if you look at the pixel intensities in a natural scene, a lot of adjacent pixels have nearly the same intensity. So their idea is that the center surround antagonism of the retinal ganglion cell receptive field will kind of cancel these kind of regions where the intensity is nearly the same everywhere. And so, what that ends up doing is it reduces the redundancy of the code at the level of the population of ganglion cells, and hence makes a more efficient neural code. So then in 1982, the first actual mention of the term predictive coding. So what Laughlin and colleagues did here is they reinterpreted the center surround inhibition of the ganglion cell receptive field by saying, because of this spatial correlation, the light intensity in the surround region partially predicts the light intensity in the center. So, so then the idea here is you take that prediction, subtract it from the actual data in the receptive field center. That gives you a different signal that, again, is a more efficient neural code. And they also applied this idea in the temporal domain to explain the biphasic impulse response of some neurons. So then in the early 90s, Attic and Redlich wrote several papers that actually quantified this principle by really calculating redundancy, at least in a model. And again, they were focusing on center surround antagonism, and they could explain changes in receptive fields over a huge range of light intensity. And the reason why there's a difference is that when you have a prediction at different signal-to-noise ratios, you'd want to subtract more or less of that prediction. So there's more subtraction at high light level at high signal-to-noise ratio and less at low. So then the last stage I want to mention is this really seminal paper by Rao and Ballard in 1999 that basically applied this idea of predictive coding to the cortical hierarchy. And so now what's happening is the higher level of the cortical hierarchy is making a prediction about what will happen next. It feeds that information down to the lower stage, and then what the brain does is it takes that prediction and it subtracts it from the bottom-up sensory data. So then what happens is only the surprising information or the errors are propagated up the cortical hierarchy. And again, the idea is this is to reduce redundancy, and this theory explains certain receptive field properties in V1. So overall, you can see in this early history that the purpose is to make a more efficient neural code. And the experimental tests of the idea are sort of all indirect, relating to receptive fields, not the prediction per se. So that brings up a big problem with efficient codes, which is that in the cortex, the codes aren't efficient at all. They appear to be massively redundant. So just as one statistic, in humans, you have about 1 million retinal ganglion cells per eye. And when you go into the primary visual cortex, per hemisphere, you have about 1 billion neurons. Okay, so 1,000-fold more neurons. There's a theorem that says you can't get any more visual information, so redundancy is very, very high. In addition, the retinal code itself is actually significantly redundant. Okay, so this whole idea of making a more efficient code, it's clearly not actually happening. Another problem with efficient codes is that they're not robust to noise. Cortex is quite noisy, so you'd want redundancy for coding fidelity. Another problem with efficient codes is once you have a perfectly efficient code, there's nothing to learn at the next stage. And so if V1 was already highly efficient, then why would there even be a cortical hierarchy? And then actually in some of our work, we've pointed out that redundancy can be critical for creating a structure in the population code that allows for easy unsupervised learning. And interestingly, not all people realize, Horace Barlow himself wrote a paper in 2001 called Redundancy Reduction Revisited, where he makes all these arguments about how redundancy is probably actually a good thing. Okay, so I'd like to maybe move towards kind of a theory of predictive coding that has more to do with movement. So the assumption here is the purpose of the neocortex is not just for creating some abstract representation of the external world, but it's there to use sensory data to make better movements. Okay, and so there are a number of problems with making good coordinated movements. There are delays in the sensory information. There are delays between the motor commands and the actual muscle contraction. But most important, in many cases, there's actually a long time for limbs and kind of the body to reach its goal location. So there are all these delays. The basic solution is that the brain is going to need to predict the sensory and motor consequences of its own movements so it can correct for these delays. But the key point I want to make here is that when the world itself is also changing, the brain has to predict future states of the world in order to make coordinated movements. And so clearly we're very good at that, and also clearly requires learning. Okay, so 2 minutes left? Okay. So I want to make a sort of quick argument about why kind of this predictive subtraction might not really make sense for in this context of making better movements. So the first thing to point out is every neocortical microcircuit has a layer of neurons in layer 5 that actually project to the spinal cord and brainstem and drive movements. In V1 in particular, they project to the superior colliculus. And so making useful movements is going to largely involve learning synapses from layer 2-3, where you have a sensory representation, to the correct layer 5 pyramidal tract neurons that will make the movement. Okay, so we should consider kind of two different types of cues for making movements. So first example, let's say you want to look at a particular face out of many possible faces. So V1 represents information about identity of faces, but that information is distributed over many, many neurons, maybe hundreds of thousands of neurons. In contrast, inferotemporal cortex has been shown to be able to decode face identity with, of order hundreds of neurons. So the idea is it's much easier and has faster learning to make this mapping from the sensory representation in IT than in V1. But another example might be making a saccade to a local feature like a mole on someone's face. And here, V1 has a compact representation of that kind of local spatial information. IT doesn't, so V1 would learn faster. So the general point is, for some movements, you want to trigger them using low-level sensory cues. So you don't want to subtract all that information away at the lower stage of the hierarchy if it's predictable. Okay, so I also want to distinguish two possible kinds of predictions that the brain is making. So the first is the case of temporal correlation in the sensory input stream, and that's what I've just been talking about. Second case is where sensory inputs are a consequence of your own movement. And so this is a sort of different circumstance where, for instance, you're going to want to distinguish something like actual motion in the world versus what seems like motion due to your own body's movement. And so here you're going to need corollary discharges, and in this case, I'm arguing it makes a lot more sense to subtract that at an early stage of the sensory pathway. So related to that, experiments involving kind of temporal correlations through repeated exposure to temporal sequences have shown predictive suppression, but at very modest amounts, maybe only about 10%. So some of Colleen's experiments, also some of my own. Experiments in the second category, where there's this kind of sensory motor coupling, manipulate the coupling between movements and stimuli. And in that case, they often see very strong suppression, maybe complete suppression. Okay, so these experimental differences argue in favor of the idea that we might want to distinguish these two types of prediction. And so in general, the brain also has different computational goals in these two cases. And so it may also want to have different mechanisms. Okay, so speaking of mechanisms, so in the first case, where there are temporal regularities, the basic idea is you're going to probably want to learn those sequences through spike time-independent plasticity. And so this is primarily driven by increases in E to E synapses. And it makes a lot of sense for this to be kind of a core learning mechanism because the spines of the neocortex take up about a third of the entire volume of neocortex. So the brain is investing huge resources in plasticity of E to E synapses. But at the same time, if you strengthen some excitatory synapses, you're going to also need to maintain EI balance. So that's going to imply you're going to have to increase certain inhibitory pathways to maintain the balance. And the tuning of inhibitory cells is broader and their axonal fan-out is broader. So that implies net inhibition when you have that EI balance restored. And so that's a possible mechanism explaining predictive suppression or novelty responses. And so in the second case, you could potentially use a negative control loop with anti-Hebbian plasticities. That's been described in the previous talks. And I'll quickly make the case argument that maybe this case is more interesting because it involves this sort of potentially more difficult problems of predicting the future. Okay, so the basic idea for an experiment to test this, I'm gonna probably skip because I'm running short on time, and I'll come back to it on a later slide. But I just want to mention, if the brain is not making predictions in order to subtract them from bottom-up sensory data, what might be the purpose? So I don't really have time to go into each of these ideas, but I just want to point out that there's a whole set of useful things that the brain can do with predictions other than subtracting them from bottom-up data. Okay, and let me skip this, but there's some experimental evidence that that this kind of excitatory process is actually happening. Okay, so in summary, what I've mentioned is I think it might be useful to distinguish two different kinds of predictive coding: the case of kind of temporal correlations in the world versus the case of sensory stimuli you've caused by your own movements. And in the first case, the sequence learning I'm proposing may be dominated by E to E plasticity. Novelty responses arise from EI balance, and I'll propose an experiment that can test that. And then just as a very last point, I think two important features we need in theories of predictive coding is it needs to be consistent with high redundancy of sensory codes. It needs to be consistent with the experimental observation that neurons, you'll find many neurons that respond reliably to hundreds of repeats of the same stimuli. Okay, so is there any time for questions or? Okay. Thank you. Okay. Well, Okay. So next up is kind of session 2, commenting on comments from experimentalists, and first talk is where's the first talk? Oh, first talk is Colleen. Oh, I'm sorry. Is this you?
Yes.
Okay.
All right. Okay, so yeah, now that we've gotten a sense of some of the big theories in predictive coding, the big question is, do we have data to back up some of these ideas? And if we don't, what experiments do we have to run next to gather that data and to test some of these ideas? What I'm going to be focusing on is evidence looking at these apical dendrites that we've been talking about a lot. And that also came up actually in the first GAC on Tuesday. Specifically, I'll be telling you about some work that we did finding that there are distinct signals in apical dendrites when you compare them to cell body populations. So just to back up a bit, as you may know, cortex has 6 layers, maybe 5 in mouse, since we call one of them layer 2-3. And one of the principal neurons that we find in cortex is this pyramidal neuron that we've been talking about, which is primarily studied in layer 5 and layer 2-3, although there are pyramidal neurons in layer 6. And so they have these cell bodies like any neuron. In their case, they're triangular, hence the name. But what's notable about them is that they also have these apical dendrites that extend up into layer 1. You'll also hear them called tough dendrites or distal dendrites, or also layer 1 dendrites. And as was brought up on Tuesday, they have these interesting properties that they're far enough from the soma that they can almost act as a separate computational unit where their inputs don't always propagate to the soma. And here you have some examples of electrophysiology recordings from Matthew Larkham. This is very pioneering and exciting work for the field that made us realize, oh, do we have 2 separate computational units in one neuron? And could they be playing distinct roles? Now, if this is the case, we'd like to know what they're doing during sensory processing. And so, fortunately, we do have some data looking at this. I'm giving just a quick overview, but there has been more research. And if I'm missing any papers, do let me know. There's a lot of papers out there, and I hate to miss one. But there have been people who have been recording in these dendrites, showing that, let's say, in primary visual cortex, they do have tuning. Sometimes that tuning will be broader than the soma that they're attached to, but there is usually a relationship. Also, the activity in apical dendrites appears to modulate the perception detection threshold. So if you have more activity in the dendrites, you can make a very subtle stimulus more detectable to an animal in a behavioral task. Now, studying these nonlinear events, the ones that Matthew Larkham and his colleagues demonstrated, these big, I'll just go right back, these big plateaus here where you get a strong depolarization in the dendrites and multiple events in the cell. It has been shown that these are more likely to occur when you have coordinated inputs to the neurons. So let's say motor and sensory input, which is in line with some of what Michael was discussing about how these sensory areas might be taking in motor information, potentially to predict what you would expect to see based on how you're moving. And also, these types of events can cause persistent plasticity. That points us towards a potential role in learning. Some more recent papers have shown that if you have an animal learning a task, there's a change in the representations that you can see encoded in the apical dendrites. Again, this is not at the soma specifically, it's at these dendrites. And also, if you suppress reward inputs to layer 1, where these apical dendrites are, you can suppress learning. So it would appear that they're well positioned to be playing an important role in learning. And of course, for the predictive coding community, the discovery of this separate compartment was very exciting. It made people think, okay, wait a second. We need prediction neurons and error neurons to be in the circuit together. What if those are actually two compartments of the same cell? So there have been a few ideas. We've heard some discussions of them. Potentially that apical dendrites could be receiving predictions and incorporating them with bottom-up signals received at the soma, somehow computing a difference between the two. Of course, a more elaborate circuit is needed because a cell can't necessarily do subtraction, but that has been sort of a pioneering idea. There's some evidence to support this already. So inputs to layer 1 do show anticipatory signals. So if an animal is used to always traversing the same environment, the axons coming in will start to anticipate those stimuli. Also, you have motor inputs coming in that carry information that can be critical for predicting visual flow if an animal is running through a corridor. And there are local interneurons, we learned from Claudia's talk, some of which will impinge upon different parts of the cell that show ramping activity in anticipation of expected stimuli. But the question, at least back when I was doing my PhD with Blake Richards in Canada, was what's occurring in the apical dendrites? And it didn't seem that very many people had recorded there. So we were fortunate to be able to do one of the first open scope projects with the Allen Institute, where we recorded in 4 different layers. This is in different mice. We recorded in layer 2, 3, and layer 5 neurons in 2 planes. Hopefully multi-plane imaging will be coming soon. At this time, it was in different animals. Here's an example of the type of data that we were able to record while animals are viewing specific stimuli. And then, we analyze the activity of the dendrites or of the cell bodies that we were able to record from. And so mice were presented with basically a sequence of Gabor images, A, B, C, D, G, which during their habituation of 6 days, they're seen every day over and over again. And then we introduce a deviant sequence that occurs only 8% of the time, where the D is swapped out for a U. And we have 3 recording sessions where we see what's going on during when they're presented with those stimuli. And what we found, if you look across sessions, top to bottom, and you see in gray the response to the expected stimulus, you see that the dendrites in layer 2, 3 are gaining a greater sensitivity to the unexpected stimulus. And this happened also in layer 5 dendrites. And so these are the dendrites that are up in layer 1. If we look at the cell bodies of the same cell type, we don't see the same pattern. If anything, we see some indications of the reverse. So we have some quantifications here. You see the increase in sensitivity in the dendrites, and that, if anything, there was a loss of sensitivity at the population level in the somata. Now, there are a lot more analyses in this paper, so do feel free to take a look at the paper. Also, the data is available online for anyone to analyze with our code and everything. So you're welcome to go and take a crack at it and find things that we didn't find in it. But just to sum up, what we found with this work was that in a passive situation, so we did have a passive task, the apical dendrites did seem to become selective to new unexpected stimuli, and this was different from what we were seeing in the cell bodies. And so this certainly points to a potential role for apical dendrites in guiding learning in a predictive coding context. Since then, there's been work from Mark Harnett's lab by Valerio Francioni showing that if you have animals doing a task, you can decode these sort of instructive signals arriving at the dendrites. They did a very clever BCI task in a retrosplenial cortex and measured the activity in apical dendrites and near the soma, and demonstrated that there appear to be signals that look like error signals being computed. But I would say that at this stage, we don't have a lot of data for apical dendrites. So if people have ideas of experiments that they'd like to see conducted so that we can really target some of these questions, there's a lot of interesting theories coming out about what the apical dendrites are doing. So we need more data. Some of my ideas are that we need experiments that really go from the start of learning to the very, very end, so that we see what happens when a stimulus is unpredictable and then when it becomes predictable. We need to be able to see the time course more closely, to see the interaction also between the broader circuit, the input axons, when they're activating, and then how the local inhibitory circuit is kicking in and how the apical dendrites are responding. And with that, I just want to acknowledge the large team that was part of this project and my co-first author, Jay Pina. And thank you very much.
Let's see. Oops.
Gave away the ending, man.
Yeah, you're going to have acknowledgments over. Now everyone knows.
Hi, everybody. Thank you for coming, and thank you to the organizing committee for setting this all up. I'm Jeff Gavornik. I'm at Boston University, and I'm going to be talking about some of our experimental work looking at sort of parameterizing the input space, or the sort of space over which we see signals that are arguably consistent with predictive coding in the mouse visual cortex. And there's, a lot of attractive reasons to think that predictive coding is something that's being implemented by the brain that have already been discussed in earlier talks. But when you actually sit down and try to do an experiment to look at a predictive signal or to see the predictions themselves, it's pretty tricky for a variety of reasons. And just to sort of illustrate this, so imagine that we have set up through some sort of training or some sort of exposure the expectation within the mouse that a visual stimulus A is going to predict a visual stimulus B, with the sort of timing as shown here, where A is on the screen or is being projected during the area indicated, well, right there. And then B comes on the screen here. AB is the predicted, and that sometimes we put in C here. And then when we put in C, we're recording from some neuropopulation. We see a response to A, and then B comes on, we see a response that looks like that. And then when we put on C, we see something that looks different than those, right? And so the question is, well, is this indicative of a predictive coding error? Is this indicative of the prediction itself? And in context of sort of how we've been talking about it earlier, is this something that you'd call a positive or a negative error? Well, it might seem like a positive error because you're seeing something you don't expect to see when C comes on the screen instead of B, but at the same time, B was expected to be seen, so maybe it's a negative error. In addition to that, if you think that it went A, C, but there was a C before this, well, we know that there are short-term accommodation effects that can modify on sort of short time scales what these things look like. And so it's not always easy to sort of tease this stuff out. And here's just another example. If we're expecting, again, with the same dynamics as in the last slide, A, B, and instead we just keep A on the screen the entire time, well, if there was an expectation that B was going to follow A, and it didn't actually happen, are we going to see something that looks like one of these red bursts that would indicate, something like a negative prediction error? And if so, when are we going to see it? Well, in order to see it at any point in time here, you have to have an expectation, not just that B is going to follow in an ordinal sense, but also kind of the temporal component and when it's expected to happen. And that sort of changes your experimental or your interpretation of what you're expecting to see, and specifically when you're expecting to see it. You need some sort of timing mechanism for a negative sort of emission like this, and maybe the type of ramping signals that were talked about by Claudia might be a mechanism, if you can tune the sort of slope of those, that you could use for timing. But anyways, it's complicated. And so what I'm going to talk about today is our use of this oddball paradigm. And we're recording in the mouse visual cortex. It's a passive viewing mouse that's awake. They're not doing any sort of task, and they're just looking at a screen which is showing repeated presentations of these type of randomly placed Gabor sequences, which are elements which we've already seen, which came from one of Michael's papers a few years ago. And the idea is that we're going to show this sequence ABCD, 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 and then every so often we're going to replace one of the elements with a novel oddball element, which is just some other random Gabor while we're recording these local field potentials. And it turns out that the local field potential is actually a really great readout of the oddball deviant response. And this is what it looks like. So the data, the blue line is the response to the standard sequence ABCD. The red line is the response to the oddball sequence AECD. So this red triangle indicates the time that the oddball was shown, that element E. And you can see that there's obvious big difference between the shape of the response generated by the oddball, and it actually persists over into the next element C, even though in context of the broader sort of structure of the sequence, C is happening exactly when you expect it to with respect to the overall timing of the ABCD sequence. And then, so the nice thing about this, you get this really robust signal. It's rapid and relatively inexpensive to do these experiments. It allows you to kind of look over a broad parameter space relatively quickly. It supports pharmacological manipulations, optogenetic manipulations to manipulate the circuit. And it has rough equivalency to the human EEG studies that have been looking at things like mismatch negativity for a while. The con is interpretability for obvious reasons. And just in terms of how I'm going to show you this data and tell you what I think is important and what is not, the statistics we're using, we just did a non-parametric hierarchical bootstrap estimate of the confidence intervals basically around these things. So what you're actually looking at here, the blue line would be the median mean over 1,000 bootstraps estimates with random replacement at both the mouse and sort of trace levels. And then 95th% confidence intervals. Our null hypothesis is that there's no difference between those two, so you do a subtraction of that bootstrap population and you get an estimate of what that difference between them looks like with those 95% confidence intervals. And anytime those confidence intervals don't include 0, that's what we interpret as being significantly different between those two at the 95% confidence level. So anyway, so that's what I'm going to show you. That's what the data looks like. And now I'm going to show you some manipulations and when we see the oddball and when we don't, and try to convince you that it's sort of more interesting than a simple passive kind of accommodation-based effect. So the first thing we did is we ran this experiment with a lot of different times for the elements, ranging from 75 milliseconds, so a very fast 75 millisecond per element, with the oddball occurring once every 3.4 seconds, up to 2-second elements with the oddball occurring something like once every 80 seconds. And what we see is that for the 75 millisecond, for the 100 millisecond, for the 200 millisecond, which is what the timing was on the previous slide, up to 1,000 milliseconds or 1 second per element, we see a nice, clear, significant signal at the time of the deviation, and then for these lower times also extending into the time of the next element, even though the period of significant difference is not contiguous between those two elements. So something is differentiating between the effect, and this sort of gives us a range over which we see the timing. So we know that there's some importance of timing. It doesn't have to be necessarily consistent timing between the elements, so we can do something where we have timing like this and we still see the oddball generating this effect. But if we completely randomize, so here each element is drawn, every presentation is drawn randomly from a distribution between 150 and 350 milliseconds, and then in that case, when you put the E, the exact same manipulation we did before, it's just the timing is now randomized, we lose the significant effect of the oddball. So the overall effect of this decreases as timing becomes less predictable. And in addition to this, there's no indication that it's actively coding for time specifically. So if you train with a fixed sequence, 200 milliseconds each, and then you manipulate the time at which the expected element is shown, that doesn't drive an oddball signature at all, which we've previously shown this manipulation does drive something that would look like an error signal in the context of this talk if you do multi-day plasticity and you're encoding this at a longer time scale. But for this oddball continuous, we don't see any indication that timing is being uniquely encoded. There is an accommodation effect, so for time reasons I'm not going to go through the details of this, but basically we show random elements with no fixed ordinal sequence, and then we plot the VEPs only as a function of how recent it was since the last time you saw the individual element that's being looked at. And if you do that, you can see an effect at the level of the VEP, which is over maybe on time scale of 200 to 400 milliseconds, something like that. You get a significant effect of this sort of accommodation. But this sort of passive, simple, exponential decay-looking accommodation can explain the complexity of this deviance response. And so the way I'm going to show this is we can basically play with the numbers, and we can show different sequences with different probabilities or different sort of frequencies of how often the deviant is shown within the overall sequence while aligning the time. So in this example, a 200 millisecond sequence where we show deviant and then 9 examples of the standard and then the deviant again. And the timing of the deviant is exactly aligned in terms of the onset with the second sequence where it's a 400 millisecond sequence, but it happens every half as often. And when we do this, if it was just a sort of passive, simple decay that was causing the difference in the signal we're looking at, you would expect to see that the deviant in these two places looks the same because it's been the exact same amount of time. We don't see that. What we see is that when we compare it, I don't have time to go through what the oddball score is, but it's just a way of sort of quantifying the magnitude of that difference. And as it becomes more surprising in terms of the frequency of the presentation, this oddball score gets larger. And this happens, so we show it for 2 different interdeviant intervals, 8 seconds and 16 seconds with these different sort of playing with these parameters. And then if you go in and you do sort of the opposite manipulation, which is hold the same frequency, so the same, once every 5, but you're changing the time, and then you go in and you plot these, you can see that once again, as the oddball becomes sort of less or more surprising by being presented less regularly within the sequence train, the magnitude of this oddball score scales in both of these cases, gets bigger with the sort of surprise metric, if you want to think about it that way. So we have other data too, which I'd be happy to talk about later if anybody wants to. But basically, in summary, we really like for this sort of investigation the local field potential. It gives you a good sort of space to explore this and constrain the models and kind of parameterize what you expect to see in terms of what you need to see to sort of match the physiology, and that these deviant responses or these oddball responses are a lot more complex in terms of how they're presenting at the level of the local field potential than you would expect for something simple like a simple accommodation model, which to me suggests that it's something that more complicated that is more in line with the predictive coding explanation. All right, that's all I have.
Hey everybody, I'm Jordan Ham. I'm at Georgia State University until October, and then I'm moving my lab to NYU and NKI in New York. So I'll just jump into it. I'm very thankful to Jeff for talking up the visual oddball paradigm. I studied it or I got into studying it basically because it has such a history in the human clinical neuroscience literature. They've been doing the oddball paradigm for decades now. And so I kind of like to say that I think understanding the oddball paradigm, and using it to study mismatch negativity, it's definitely benefited by applying the predictive coding framework to it. I don't necessarily think it does a whole lot for understanding predictive coding. I think it's a special case. It can be useful for studying predictive coding, but it only kind of cuts out a small chunk in the story. But basically, to summarize what it is, we just show the mouse the same visual stimulus over and over and over until it becomes redundant, and then we show a different orientation. And this is primary visual cortex, and you can see on a single trial in individual neurons, you can see the deviant detection response. It's huge. It's a really large magnitude. So we actually, in my lab for years now, we've been running the same 3 paradigms, same 3 sequences. We essentially do a basic visual oddball paradigm, and these are moving gradings. They weren't in the previous slide, but just trust me. You show the same orientation over and over, and then you show a different orientation that's deviant. Then we do a flip, so then what was the deviant before is now the redundant stimulus. And then we do many standard frequency control. So in this case, we essentially show the mouse any sort of random order of many different orientations, such that any given orientation is still equally rare as it was during the oddball paradigm, but it's not contextually deviant because it's in the context of any orientation goes. And so we essentially are able to compare responses to the same orientation in the same mice when that stimulus is kind of contextually redundant versus when it's contextually deviant versus when it's contextually equally probable in a selection of many other different orientations. And so these are the average responses of V1 neurons in layer 2-3 to the same stimulus across these three contexts. And so you can see, you do see an adaptation effect, meaning you do see reduced responses to that stimulus when it's being repeated once every second over and over. And we call this stimulus-specific adaptation. But you also see an increased response relative to the control, which we call deviance detection. And in the context of this GAC, we're calling it kind of a prediction error. I like to think of it as a kind of a rudimentary form of visual prediction errors in this oddball paradigm. And we've shown this with the same paradigm multiple times now. A couple interesting things about deviance detection, a couple interesting things about this kind of basic visual perceptual prediction error is that it's enriched in layer 2-3 of primary visual cortex. Here we just did multi-layer calcium imaging in mice, and we see in layer 4 and in layer 5, determined mostly on depth, you see clear stimulus-specific adaptation across all these layers. That's there across all the layers. But this deviance detection response is really only up there in layer 2-3, up there at the top. And we replicated this with a multi-electrode recording looking at multi-unit activity, so just doing ephys, and we see essentially the same thing. We really only get this deviance detection response up there in layer 2-3. The stimulus-specific adaptation is not only spatially distinct from your deviance detection response, but it's also temporally distinct. We see it very early in your layer 4 responses. And actually doing current source density, we even see it in the early currents that we are assuming are coming from LGN. So it's spatiotemporally distinct, these two mechanisms. So I really like to say, as Jeff suggested, the deviance detection response in the oddball paradigm is still pretty interesting. It's more than just the absence of adaptation. And I think it's worthwhile to study. And so can we kind of make this kind of labor on the assumption it does represent some sort of prediction error. So I'll go over some of our experimental data. It looks like a model. It's just me drawing out my hypothesis. I'm not a computational neuroscientist. There's nothing kind of in any quantitative way that I have to represent this, but this is just kind of for structuring our experiments. We've seen so far that you have both adaptation and deviance detection in layer 2-3 in pyramidal cells. But what about these other inhibitory neurons that have been brought up so far? And also, what about top-down inputs to visual cortex? Well, we did calcium imaging of pyramidal cells, somatostatin-positive interneurons, VIP-positive interneurons, and axons from anterior cingulate area, which is an area of mouse prefrontal cortex that projects heavily and directly to V1 in layer 1. And we looked and saw what types of signals are present in these neurons. In your somatostatin-positive interneurons, you don't have deviance detection, but you have stimulus-specific adaptation. In your VIPs, you don't have deviance detection as strong. There's a little bit there, but you definitely have a very strong inverse adaptation in a subset of these cells. And your layer 1 axons don't really show a huge effect. This is just the same data. I've just plotted them for the same cells over the same axis. This is asking which cells are there deviance detection in, and it looks like you only have it in your pyramidal neurons, and it's relatively early. But which cells do you have adaptation in? Well, you have stimulus-specific adaptation at both in SSTs and pyramidal cells, and it might actually be stronger in SSTs. And while VIPs show inverse adaptation, they go higher than they were in the control case. But what about your top-down inputs? They don't seem to be doing anything all that interesting. Maybe this isn't where the prediction is coming from in this paradigm. But wait, so we did local field potential recordings, which I also happen to think are very useful, very cool, and pretty cheap. We did recorded from V1 and prefrontal cortex and ACA in the same mice, and we quantified synchrony between these areas. And you can see that the synchrony during the oddball paradigm sort of peaked in this theta alpha range. And we did Granger causality looking to see how activity in and ACA influenced activity in V1 and then vice versa, because these regions are bidirectionally connected. And what was interesting is during the control paradigm, that dark gray versus light gray, that suggests that the causality or at least the influence of one region on another is balanced. There's just as much bottom-up and top-down influence going on. But during the redundant stimuli, ACA, that left bar, seemed to dominate the relationship. So ACA seemed to be driving V1. And you almost start to see the inverse thing happen during the deviant. So it definitely suggests that there's some interesting communication going on between these areas during the oddball paradigm. So what we did is we stimulated optogenetically these top-down inputs with channel rhodopsin at the driving frequency, at the 6 to 10 hertz range that we found was interesting in the paradigm. And we measured, so this is driving ACA axons in V1 while measuring VIPs, SSTs, and pyramidal cells in different mice. And we found is in this 6 to 10 hertz range, the VIPs loved that stimulation. They go way up. And the somatostatin neurons actually show some decrease, at least as far as we can tell from calcium signals. So it's not perfect, but it at least suggests that their calcium signals were below what they were at baseline. So it looks like stimulating this circuit in this particular frequency range, which seemed to be important for the oddball paradigm, actually activates VIPs, sends them way through the roof, and inhibits SSTs. And again, I'll just have you recall what the calcium imaging data looked like during the oddball paradigm. Exactly the same thing. VIPs went up, SSTs went down. So we're starting to put together a picture of what the communication between ACA and V1 might be during the oddball paradigm. The other point is what kind of information is being sent. We know what it looks like in the time domain. We don't really know what it looks like in the spatial domain. So we image these axons in V1, in layer 1 of V1, injected GCamp and ACA and imaged the inputs. This is just to kind of depict what our analysis was. We looked to decode the stimulus ID from the activity. This is pyramidal cells in V1, so I'm taking a step back. You can very clearly kind of decode what orientation the mouse saw from the pyramidal cells in V1, as expected. When the mouse saw 45 degrees, you see that kind of trajectory goes up, and when it saw 135 degrees, it went the other direction. If we did the same thing with ACA axons, you can decode the orientation in the oddball paradigm when it's redundant. And you can decode it when it's deviant, but it actually seemed to go in different directions. And at first we were like, why would it go in different directions? And the reason is that the information being sent, at least this is one interpretation, you can kind of see the dark dots on the left go down. That's when the trials when the mouse saw 135 degrees, and they kind of go in the up direction on the right. The suggestion is that ACA isn't sending information about the current stimulus to V1. It's sending information about the expected current stimulus to V1. So when the mouse saw 135 degrees, it actually expected 45 degrees. And this is why the trials, when the mouse saw 135 degrees, when it's deviant, it actually might have, the ACA inputs were way different. And then we were unable to discern or to decode the orientation during the control paradigm, suggesting that ACA doesn't know what orientation the mouse is about to see. So the axon information being sent back doesn't contain any orientation information. Okay. And just to kind of wrap it up, we have gone through and inhibited each one of these cell types and circuit legs, and every time we mess with them, it messes with deviance detection. So at first we were like, yes, this is necessary. And I'm starting to think if you mess with the brain, it messes up deviance detection. So just to point out, in the papers we've done this, we kind of inhibited ACA inputs to V1 with ARCH-T. It eliminates deviance detection in V1. Interestingly, it doesn't mess with stimulus-specific adaptation, consistent with the notion that this is an inherited adaptation from bottom-up sources. Same thing, we inhibited VIP interneurons using DREADS. And in this study, we were using LFP, and we saw deviance detection eliminated. Adaptation wasn't messed with. Interestingly, though, messing with VIPs decreased coherence between ACA and V1. So we lost that communication point, consistent with the notion that the VIPs are mediating that top-down input. And then finally, we suppressed somatocyte interneurons and we lost deviance detection. We didn't lose stimule-specific adaptation. So any one of these elements appears to be necessary for the picture, but how they interact, I think, is the more important thing for predictive coding. Okay, so I'm going to end with that. I kind of had an explanation of how I think all of this goes together, but I don't want to run over too much. This is just to say I think this is, I've done a very deep dive into a very limited paradigm. So whether or not this is going to apply to a more complex oddball paradigm, whether it applies to kind of a corollary discharge, I wouldn't bet on it. But I think it explains some of the oddball mismatch negativity P300 type of data pretty well. Okay, and this is my lab. There's quite a lot of them. But none of them are coming to New York with me, so if anybody's interested in a postdoc, I'm all ears. Thank you.
It's just the browser. Yeah.
Let's open that up again.
This one.
All right, actually, let's just present. Okay, so those were the talks, and now we're going to move on to the discussion. Before we jump in, I do want to just remind everyone that if you follow the QR code, you can go take a look at the position paper and find out about how to contribute to it if you're interested. We will now move on to presenting very briefly a couple of proposals of experiments that we might be interested in running before opening up the floor for everyone else to contribute to the conversation.
All right, so in the document you can see there are 3 groups of proposals. We try to group them a little bit, and so there might be more of it coming. There is a group of proposals on the diversity of mismatch mechanisms. You've seen that the field has used different kind of mismatch, and so there could be interest in characterizing the different mechanisms at play. Another group of proposals is probably model-driven. So you've seen some of the theory that has been advanced. And so we could test some of these ideas if we think as a group is important. And the last one is probably about functional anatomy. So like, what are the different layers doing? What are the different individual neurons? I like to propose a first start. And so you've seen in the talks that there are different mechanisms at play, from adaptation to short-term memory. There might be figure ground involved in surround suppression, mismatch type of experiments. And you'll also talk about corollary discharge. And so predictive coding is assumed there is a single mechanism at play. But there could be several. And so what I'm proposing here is that we try to design an experiment that collects the different kinds of mismatch to build together a standardized data set that will allow the community to test different models against one another. And so the way we do this is leverage the 2-photon platform at the Institute, as well as the NeuroPixel platform, and design a stimulus that contain A, B, C, and D, and collect in separate mice 2-photon with like wide surveys of inhibitory and excitatory neurons in the same cortical column, and do the same with NeuroPixel recording. So you benefit from the strength of both technology, and the field designed this standardized stimulus set. So that's my proposal. I'm going to hand it out for Colleen.
All right, so a second proposal, I won't spend too much time on this because I think I've already sort of explained it partially in the presentation I gave earlier, but the idea would be to delve into what signals we find in layer 1. And so, if we're able to record in multiple different channels, it would be great to be able to see what's happening not only in the apical dendrites, as in the experiment I discussed earlier, but also in the axons coming in, so similar to the work that the Ham Lab's been doing, and ideally also in some of the interneurons that are in layer 1 that we suspect are involved in carrying these signals. For example, NDNF neurons that are actually in layer 1, or inhibitory neurons that reach into layer 1 onto the apical trunk like somatostatin. And the idea with this kind of experiment would be to really cover the full range of learning, as I had mentioned earlier. So initially habituating an animal to a predictable stimulus, a stimulus that follows some rule A, and capturing the activity as it's habituated to this stimulus, introducing unpredicted stimuli that follow a different rule, one that can be learned, recording the activity throughout the entire learning process until we judge that the animal has been habituated to this new stimulus and it is in some way predictable for the animal. So seeing the full cycle of integrating new information into the internal model that we think is being learned within the predictive coding framework. So that would be a second proposal. And then Michael, you have a third one. I'll hand it off.
Okay, great. So yeah, so the proposal I want to make has to do with this this question of whether the E to E pathways are actually strengthening in predictive coding paradigms. So the basic idea is to kind of run a multi-day experiment that kind of doing a predictive coding paradigm. So the basic idea would be expose the animal passively to a temporal sequence of stimuli. And so over multiple days, the temporal sequence, let's say it goes A, B, C, D. When the animal sees stimulus C, it potentially learns over that time period that the next stimulus will be D. Okay, so the idea is you'll look at kind of responses of a population of neurons before and after training. So this is kind of a representation of these sparse codes where it's a heat map. So kind of the hotter colors are more active neurons. Black is not active. And so I'm just showing here an example of a type of experiment we've done in my lab where you do this kind of temporal sequence training over multiple days. And what you see in the population activity after training is that some of the strongly active cells remain a similar activity level, but the moderately active cells are suppressed. So there's kind of a net suppression, which is consistent with predictive suppression in a lot of theories of predictive coding. But so then the idea is if we think the E to E pathways are making a contribution to this, then let's block inhibition and see what happens in that case. So this is kind of a picture of what might happen after training if we're only looking at if we've blocked inhibition. The idea is the specific network of cells that are excited by the stimulus. In the predictable case, their activity will be enhanced. If there's only inhibitory plasticity, then nothing would change when you block inhibition. And then one should include some other important controls, like you can show this stimulus without the preceding predictable context. Okay, what happens there? You can also show a sorry, sorry. Just showing a new stimulus to see if there's some kind of overall differences in excitability. So one would want to include some other types of controls, but the basic idea is block inhibition, see what happens to the E to E pathway.
So I think now we want to engage with you. And so I might start with questions, and then the audience, I'm hoping that you engage. So remember that we are designing an experiment. We have committed funds, so we're going to run an experiment. It's going to happen. So I'm going to start with a question to Jordan. What do you think is going to happen in these experiments? Because you've said that inhibitory neurons matters.
Yeah, but then that's all.
Maybe take a minute.
Okay. So I don't know, but I think it will be. I think it'll be informative. I guess it sort of depends on how we block inhibition and whether we can block inhibitory plasticity and or inhibition from specific cell types. But again, I think the thing that sticks out to me, and has come up a few times, is that I think predictive coding or predictive coding mechanisms may be different, or even may be best studied separately as sort of ones that, kind of on long scales and short-term scales. Either sort of short-term predictive coding might not lead to as much plasticity. I mean, and I can see it in my paradigms because I run one oddball paradigm and then 30 seconds later I run the other one, and I'm still getting deviance detection. So the mice didn't learn too much, or at least if they did, they unlearn it very quickly. Whereas, I think some of the stuff that he brought up, predictive coding should at least have some pretty dramatic effects on plasticity when they're learning a kind of task that predictions actually mean something for whether or not they're going to get a reward or not. That means something about how to change their behavior. So it's possible that there are some mechanisms that apply to a paradigm like that might not apply to the very short-term paradigms that like Jeff and I study. And I guess that's my take.
Any other comments in the audience?
Yeah, so I would add also that, I think the oddball paradigm, like I talked about today, is fundamentally different at a mechanistic level from the multi-day plasticity that I've looked at before, where you show something that sounds very similar to this proposed experiment of, a fixed sequence of gratings or a fixed sequence of whatever that you show over multiple days. And that leads to big changes in the local field potential. It leads to changes in the dynamic firing rate of individual neurons. But it is not blocked, at least in the experiments that we've done, it's not blocked by like CPP, which is a, arguably, I don't know what you were thinking about using for this, but blocks E to E plasticity in the kind of classic Hebbian sense. That didn't have any real effect on the sequence plasticity, whereas blocking the muscarinic acetylcholine receptors with scopolamine did. And I don't know what that means as it relates exactly, because and we think it's actually the M2 receptors specifically that are important for that. But I don't know how that really relates to specific plasticity at specific locations within the network, because no one has really characterized a good relationship between those particular receptors and any of the individual subtypes within the network. So the other comment I would make as it relates to just sort of low-level experimental design is that when we do that oddball paradigm, if we show the stimulus continuously, like I showed in the talk today, we see very minimal changes in evoked potentials over multiple days of that same stimulus. Whereas if we do ABCD and then we have a long gray interstitial and then repeat ABCD, that drives much larger plasticity. For what reason, I don't know. But it's something to think about in terms of the design, and that differentiates that kind of short-term oddball deviance response from a more long-term coding of the spatial temporal expectations that are defined by the stimulus you're using for the training. And I mentioned this just briefly in passing, but we do, the multi-day stuff does seem to be much more specific for the unique timing. So if we do an experiment where we show AB with a particular timing and then we hold A on the screen, you see a deviance response or an error response generated at the time that B would have come on the screen, even though the stimulus doesn't change at all. So.
A quick question. We have seen large effects of the task, and I haven't heard so far predictions of how the task would contribute to the prediction error. Obviously, it's important because it might be more or less meaningful in the context of what the mice are doing. So maybe because of the briefness of time, you didn't mention it, but maybe you can comment on it now.
You're asking about the behavior task, or?
Yeah, behavior. So in the experimental data for the in Ham's talk, for example, for the sequence, did you see effects of task specificity of the mice, whether they were, or what was the task? Were they fixating? Were they responding? Were they ignoring the stimulus? Or
they weren't doing anything. It was passive. Yeah, thank you. Yeah, it was passive. I actually. I mean, I modeled it mainly after the human oddball mismatch negativity studies, because those were always passive. So the idea was to figure out what's driving a mismatch negativity. So intentionally, there was no task, and you still get this large response. I do think that, and Jerome's work, that's been published, in those cases, mice were trained to respond to omissions.
So in some papers that we did not show today, we trained mice in the detection of change task, and as part of the stimuli, there are omissions, so you could say negative error. And there we see very strong VIP engagement to the omission. So the mouse is attending, but it's not part of the task, like the reward is a side product, yeah.
But that actually led to, those data are very different than what we've seen in the passive oddball task. And so it does bring up the point that maybe having a task there could very much change the responsivity of V1.
It's possible, yeah. So.
Hi there. I want to continue on exactly this thing because I reacted to the name of the session, Attention to Prediction Error, and attention has not been discussed but alluded to at several points now. And I actually had exactly the same comment as this previous commentator, that these kind of prediction error responses, at least when you look at like the higher level, like EEG responses, we've been discussing mismatch negativity, which occurs both in passive and active situations, although it's much stronger when you are actually attending to the stimuli. But then there are several that don't appear unless you're not attending to the stimuli. In the human literature, you would, for example, talk about the N400 response and so on. And there's also theories that suggest that attention works by like making the prediction error responses stronger, like putting more gain on this. So I think there would be really interesting things to look at, like on the cellular level now, but that would mean that you also would have to think about the tasks and the type of what the animal is attending to, and maybe have a situation where you're not attending to anything or superimposed, and then like change the prediction, stimulus sequences and so on.
I can comment on that. So in the perspective piece, well, thanks for the contribution from the community. We started to have a long section of discussing attention, and especially the part that you mentioned, the predictive coding aspects to this, which is like the saliency and the weight and the prediction error. I think my understanding is mice work don't consider that too seriously. I think it's a lot of the human literature. To do this in mice, I think we might have, I think some people suggested in the piece to do distractor stimuli to sort of move attention away. So this is something we could do. I'm curious if other people have ideas here.
Well, I think actually it fits kind of nicely into your idea of looking at different types of error types and sort of building that data set. If we could have experiments that are as similar as possible on most fronts, but differ on the attention front, then we could get to that question, because it comes up a lot. You know, having published a passive paper, that was a big question, was why do you even think the mice are paying attention, and do you think that this generalizes to when there's a task? So yeah, I think that's a really good point that we should make a note of. Thank you.
Hi, nice stuff. I just skimmed the position paper, so I mean, I guess I may be a little bit naive in a couple of my questions, but I note that, I guess like the traditional forms of predictive coding, as was alluded to when you think of Barlow and the ideas of its relationship to efficient coding and so on, it has to be predictions within a spatiotemporal statistical context. So for the retina, you have natural statistics of the world, which have correlation structures that allow you to imbue that from development, on an evolutionary time scale. In prefrontal cortex, you may be paying attention to completely different time scales that are learned in the animal's local lifetime. And the prediction mechanisms at definitely the implementation level may be very different. So I'm thinking, for example, it's not clear that even V1 to prefrontal cortex would necessarily want to be predicting on the same timescales in the same way, and therefore the microcircuitry shouldn't necessarily be the same. And so first question is broadly, how are you thinking about that? Because right now it's very hard to think about what experiments to do without having a kind of more concrete perspective on how we're actually thinking about predictive coding. And I guess a related thing that I've always struggled with is, in a lot of the kinds of evidence that we have, the really scalar prediction errors that we're seeing, which I'm coming from an RL background, so I love scalar prediction errors. But actually for things in V1, the expectations and the predictions are all vectored objects, right? They're high dimensional. And without being able to actually know that what you're looking at is really a prediction about a particular object, it's very hard for me to know whether they're not compatible with a whole bunch of other theories which might not involve prediction error at all. So I guess those are two broad questions to start with.
I can comment on the first one. I think there's a proposal in the document. I really encourage you to take a look. That is proposing to change the temporal dynamic of the sequence. So you can change the frequency of the flashes and study whether like V1 has a certain temporal structure that is different than other areas. So we could do that. On the modeling aspect, maybe Fabian, could you wait on that?
I mean, I can comment to the second question a bit. From our perspective, of course, and we don't have these error neurons. So our theory would expect, to some extent, that a lot of these responses come from other processes, like attention processes, for example. And then you would, of course, have different dimensionality, let's say, as you phrased it, in the error response. And there are experiments that also look at that, but I actually only know one which still has to be published. But this finds that this response actually looks more like a boosted sensory response and less like a specific error response that is specific to the precise error that is shown. And so this is definitely something that I should think that should be tested more directly and more investigated more, because that would distinguish between these two very broad categories of mismatch responses, which are like attention-like responses or error-like responses. Yeah. So, yeah.
I can just add to that there was one paper that I think was trying to get at what you were saying, and that was the Francioni paper, where the idea was, okay, we don't know what the prediction should look like because we are in the brain, so let's create a task where there's a BCI, and therefore we know what signal the cell should be receiving. So that can be a way to approach the question, I suppose. It's hard to train a BCI on V1, unfortunately, as we found out from that team. They ended up in retrosplenial cortex. But yeah, that is a real challenge. We don't know exactly. We know sometimes, okay, these are B stimulus responding neurons, but we don't tend to have sort of a high dimensional signal that we can look for. And I think that's an important point.
Cool, thanks. I just want to thank everyone for, thank the organizers and thank the speakers. This has been a really thought-provoking and super interesting symposium. Thanks for putting it together. I just wanted to play a little bit off what Eric said, and when he brought up high dimensionality, I am very sympathetic to the excitatory plasticity ideas, just because of the really high dimensionality that exists there, right? You have, something like 80% of the synapses on a given pyramidal cell coming from the local neighborhood, and that's like of like 1,000 at least inputs. And so you have something like 1 billion excitatory synapses in like a cubic millimeter of the cortex. And that is potentially like a really high dimensional, kind of a learning substrate. So I think like exploring these kind of excitatory interactions is to me super interesting. And then I guess the second point I'd make is, relating to some of the dendritic proposals that are very interesting and the dendritic data that are shown. This kind of came up in the position paper, but in the comments. But there's, a traditional concept of predictive coding, and sort of what that evokes is like error signals and sort of differences in oddball responses. But I think I liked what people were saying about kind of predictive processing more generally, the idea that we don't necessarily need to have an explicit error signal. I mean, you can imagine, of course, in the dendrites that there could be some, as just an example, some kind of active process that is triggered only when a certain conjunction of synapses arrives. And that is the kind of thing that wouldn't necessarily, you may not even see an error signal, right? It could just be sort of some kind of like cryptic predictive processing. So on both those ideas, I wonder what the panel thinks.
I have some thoughts on that because I remember during my PhD it came up a lot, this idea of predictive coding, and it seemed to be what people attributed most of the data to when we saw predictive signals. But presumably there are many other ways of predicting about the world that don't look exactly like predictive coding. And it seemed to me that what we were missing were a couple of fairly concrete parallel models that we could really compare to say, okay, what do they predict? Of course, there's always limitations to that because all the models are going to be incorrect, and you don't know if the way that they're incorrect critically change the result or is just incidental. But yeah, I think in some ways having this position paper focused on predictive coding has some advantages in the sense of saying, well, we want to specifically test if this is the algorithm that the brain is using. And then a lot of the ideas brought up can still be reused in the direction of alternative models that might break onto the scene. But I agree that as scientists, we should be thinking more broadly and not when we see predictive signals, just thinking it's predictive coding, but it's potentially in the class of predictive processing models. Those are my thoughts on that.
If you look at the perspective piece, there seems to be a pattern where the temporal dynamics involve a lot of layer 2-3, maybe recurrence. And the sensory motor is really heavy on the inhibitory signaling. And so I wonder if there are two distinct phenomena here, and maybe one is actually using E to I balance in the context of sensory motor pathway, and it's like feedback inhibition essentially. And other context where the layer 2-3 is just taking over and doing that, so sort of explain away. Does that make sense? I feel that's why I think we should acquire data where we can disentangle the different mechanisms in the same mouse, because I think that will move the field forward. And I think it would be nice if multiple labs collaborate there. So like if we could have the same experiment run across different labs, perhaps with different modalities, so that we complement one another. I would love to see dendritic imaging with the same kind of stimulus, for instance. We saw complex experiments. It would be better if it's the same stim as the survey. So that's my thought. I just wonder if other people have ideas there.
In different species.
Well, the different species, yeah, we're already doing some of this with Androbastos. It's going to get out, but it would be great if there's a little more, yeah.
Hi, guys. Thanks so much for your talks. One of the points that I found very interesting that was brought up was about the role of efficient coding in making these predictions. So, my mental model often when I'm reading papers about predictive coding is, well, if I was a brain and I was lazy, I didn't want to use energy, then I may as well compute what I can. I may as well predict what I can so that when the time comes, I don't have to do the work and I can just skip to the point. I was wondering what the other speakers thought about the role of efficiency and how that plays into the way that you guys think about predictive coding as you run your experiments or do your theory. Yeah, really curious to what you guys think about that.
Yeah, I mean, I guess I should say something about that. I mean, so I was arguing, based on empirical evidence that, the cortex is actually not very efficient at all. And, so I guess one perspective on that is that, there's some value to redundancy in sensory codes or just, information coding in general. Such that you're going to always want to have that. Another way of thinking about it is, let's say that value of redundancy imposes certain constraints, or there are other, let's say, computations you just absolutely want to do. Given those constraints, you could still have a more efficient, computation or code, than not. Once you've kind of put in as a constraint, let's say you want to have a certain amount of redundancy for various reasons, So some of these kind of subtractive versions of predictive coding, could, you could, let's say, still be, creating a somewhat more efficient code than if they weren't operating, even though there's this constraint in the system for a lot of redundancy overall.
I have some intuition that I want to share. It looks like the cortex is expanding the space because it's an expansion. So it's probably doing this to explore let's say, the space of possible output, and then further constrict it down the line. So it's parallelizing the exploration. And so I think it makes sense that in a cortical column there's part of it that is expanding. And then as it goes down, it will restrict down. I think in the perspective piece, we comment a lot on the role of parvalbumin cells as doing a little bit of that true EI balance, or filtering out some of the pyramidal cell output. There's a lot of work that has shown that actually that happens, through inhibitions. And so it's possible that as I said, there are multiple computation happening in the cortical column, and so some of this could be predictive coding-like, and some of this could be explaining away that involves certain subtypes. It's a part of the challenge, right? Because it's tightly integrated, and so we need to design experiment that disentangle these different computations.
I just wanted to mention something like you, but thank you.
Please. You agree?
So I like the idea of efficient coding for a variety of reasons. One of them is because it makes sense sort of in an evolutionary perspective, right? That you want to make your brain more efficient so you don't have to use as much energy or as much whatever. But the thing that's always sort of confusing about it to me is you don't really know what the definition of efficiency is that's important from an evolutionary time scale, right? It's not necessarily the number of action potentials. It's not necessarily the number of cells that are representing any particular object or idea. It could be a lot of different things. And so it's hard to, and I think sometimes, especially coming from like the engineering perspective of, information theory, we sort of look at it and we say, we're like, oh, this doesn't look efficient in the, in the sense that like it's using 2 more bits than it should if it was actually optimized. And it's not necessarily optimized in that sense, but that doesn't mean it doesn't give you some intuition. And the other thing that I really like about the ideas of efficient coding as it relates to these kind of sequence models is that, you have redundancy, or you get a better functioning code by reducing redundancy if you include the temporal component built into the code. So a predictive code is more efficient than a non-predictive code if you have spatial regularities in your sort of, statistics of your input space. And because I'm really interested in the kind of the relationship of the representation of time in these networks, that to me sort of argues as this is a good way to, to create codes that are efficient, and in doing so also give you the ability to make predictions based on the statistics of the learning set. To me, that makes a lot of sense. So it's hard to know what to expect that to look like in a biological sense.
I think that's our time. If you'll give me just one moment, I know that the intro was not live streamed, so I'll just remind everyone that We do have this open scope project that we're looking to design, and that will be a go. It will be supported by the Allen Institute and the NIH, and the data will be put up on Dandy after. Also, make sure to join on our proposal, our perspective piece, if you have any expertise or interest in the field. And yeah, if you want to join us, we're going to Nako Tako. So yeah, thank you everyone for your attention and contributions.